next speaker is Yanlan Mao. So she's from the LMCB at UCL, and she knows David from her time as a postdoc in Nick Tapon's lab in the LRI. And the title of her talk is Coping with Mechanical Stress, How to Build Robust Architecture.
Great, thank you. So first of all, thank you to Roz and the other organizers for this organizing this amazing day and for scheduling me after Paul. It's always a hard act to follow. I'll try my best. And so David, so I was never actually officially in David's lab or an official mentee, but we actually shared 15 years together. We shared the same building at the LRI, which I think no longer exists, this beautiful building there. But then also after that, we both moved to the LMCB at UCL. I'm not sure who followed who, but anyway, we were then there for pretty much 10 years. And we shared lots of coffees and lunches and Christmas parties and cocktail parties, the famous LMCB cocktail parties. And yeah, I mean, as you all know, David really shares a lot of passion. Many I also share, for example, food, especially noodles, and there are lots of great noodle bars around UCL that we would, go to. But also, of course, travel, skiing. This actually isn't him. I couldn't find one of him, unfortunately, so I just sort of grabbed one remotely. And films. He was always fascinated by different films, and this one, Tampopo, again, see the common theme there. And as you all know, he's incredibly passionate about really everything. He knows, deeply about everything, and so we shared a lot of conversations. But I think, and again, I'm sure amongst you all share many things with David. But I think one thing that's quite unique that I share with David is our love for architecture, or rather, our love for architects. So our better halves are both architects, and John actually picked this. So John's my husband, who's the architect, actually picked this picture. I was going to pick like Eiffel Tower, really cheesy, really boring, but he said use this because Roz will appreciate this drawing, and the scientists among you maybe we can discuss this afterwards because I wasn't really anyway. So yes, also therefore during our coffees and lunches we would talk a lot about architecture, the Venice Biennale or whatever, and of course also he would tell me about how the crick wasn't quite what he wanted and how the stairs were not where he wanted it to be and so on and so forth. So lots of discussions, I think. So yeah, I think he really liked talking about architecture to scientists, in these scientific environments. So that was pretty unique and something I really enjoyed. So of course, we all know it's really important to build really robust architecture, right? and hopefully, and even here, sometimes you can feel it, right, when the trains go by. So it's really, really critical to build robust architecture that's resistant to environmental fluctuations, mechanical forces, such as earthquakes. And obviously, there's also lots of diverse and very beautiful architecture in our kind of physical world. And that really applies to biological architecture as well. Biological architecture, form, function, tightly linked, very beautiful, very diverse. And Ilan, by the way, this is the picture I was telling you about in Glasgow. I pretty much grew up in Glasgow, and this Kelvin Grove Art Gallery is, I think, close to where Ilan lives now. And this photo I actually took from there. And this is using my phone from really ages ago, so it's really rubbish pictured, but I always use it because it says pretty much exactly what I work on and why I'm interested in science, which is how do you get this beautiful diversity of shapes and sizes and form. Even at the very basic level, we're built up of very similar building blocks. And I saw this when I took my daughter actually to visit grandparents to see this museum that I grew up going to see every day, or every weekend at least. And this display was just there. And I said, okay, this is, I have to take a photo of this. So it's sort of stayed in my iPhone and I use it regularly. But essentially, yes, biological building or biological form has to be very robust and it's very diverse. And we actually develop in constantly fluctuating mechanical environments, right? We might think we grow in very sheltered, very protected environments, but if you're a plant, it's pretty obvious that it's most of the forces, in fact, the only forces really experience this is from the external environment. And if you keep poking a plant, keep touching it, it's going to really affect its growth, right? So that's kind of clear. And actually, it's the same for animal development, even fetuses. You may think we're very protected in our mother's wombs, but in fact, the motion from the mother, in fact, the kicking motion of the fetus itself, this generates stochastic mechanical forces that are basically unpredictable and therefore, by definition, fluctuating. And these will actually affect the development of the fetus. And it's the same for developing the heart. Pretty much as soon as the heart is, the small valves are formed, it starts to pump, and it's basically developing as it's moving, as the functions as the forces are fluctuating. So it's this kind of constant interplay. In a way, we are definitely sensitive to mechanics, we know that, but how do we still robustly develop and actually create this perfect form, even though we have to develop in these constantly fluctuating environments? So that's kind of the conundrum we're trying to understand now in my lab. And it's not just about development, but it's also about homeostasis, because after we've developed, we then need to spend pretty much the rest of our lives trying to stay in this form. And so, of course, it's not as if the forces around us stop. All of us here, our hearts are beating at different rates. Mine is probably much faster than yours right now. Or I suddenly go for a run. So these, again, are stochastic, by definition, unpredictable. They are not genetically encoded per se, right? Yet, most of us can tolerate these fluctuations. But of course, there are also extreme cases when if you have too much force, so for example, athletes that are constantly, overloading their hearts with extreme exercise, their hearts do undergo hypertrophy. So again, it's not because we're insensitive to these forces, but somehow there are ranges where this can be tolerated, and then there are extremes where it becomes pathogenic. So what is this tolerable but variable range? And how do we know how much is too much? And maybe this is actually different for all of us individuals. As individuals, I think we have tolerances to different external mechanical insults or stresses, and not just mechanical, but any external stresses. So, but as you can see, the theme in the talks seems to be all about mechanics. And so you saw stretching devices from Helen earlier, and also we've been basically designing different ways, essentially, to apply controlled mechanical forces to a tissue. Mostly we work on whole mount tissues, and then observe, or at least quantify the behavior of these tissues to try to understand some of these questions I've been kind of posing just now. And so, for example, we built a tissue stretcher. Essentially, you can basically clamp your favorite organ of interest in between two layers of PDMS, and you can culture it in its kind of perfect media. And then you can apply the whole device on top of an inverted microscope. So you can image the response of the stretch, and you can stretch it by either rotating this motor clockwise or anti-clockwise. And you can also apply different cyclic perturbations, different cyclic stretches, so different strain frequencies, rates, and extents, okay? And so, for example, we noticed that if you were to apply this stretch, and this is while we're imaging live, by the way, the response, so you can use different markers, and this is mostly in fly tissues, and it allows you to observe very quickly, very dynamically, how the cells respond to the stress. So this is published years ago, but long story short, essentially what we saw is that, like many others have now seen, that actomyosin cortex actually polarizes within minutes in response to this mechanical stress. And effectively what that does is it stiffens the tissue to prevent it from further deformation. So this is a very rapid protective response. And I guess it makes sense. If someone's trying to stretch you apart, you want to fight back, you want to resist that deformation. And so this preserves the shape of these organs, at least in a minute's time scale, to prevent them from constantly fluctuating and moving when there are these natural fluctuations in their deformation. Okay, so that's one kind of mechanical response. And so these external kind of structures are really useful for understanding this kind of high resolution cellular level responses. But of course, the problem with this is that after you cut the wing discs out, at least I don't know how to put them back into the larvae again and let them develop. I mean, I know some people can, but I can't. And after all this kind of stretching, it's really hard to then follow the development of these wing discs and look at the long-term consequences of the stretch. And so what we are doing now is actually trying to understand this in vivo. So you'll see, again, this exemplifies the fact that we're never, or at least the larvae, are never developing in a static environment. So the normal development of a larvae with all the organs, the wing discs and so on embedded inside, they're actually constantly undergoing mechanical fluctuations, even during eating and crawling of the larvae. And so, for example, what we're doing now is to actually image and quantify these fluctuations. So this is just the wing discs of the larvae highlighted. And we confine them slightly in these channels to allow us to kind of image them at high temporal resolution. And you can see the organs are very much fluctuating even during normal development. And so what's kind of interesting is every wing disc will experience different forces, yet every wing in a wild-type scenario will hatch out to be pretty much exactly the same size and shape. So it's incredibly robust development. This is why we use wings as for genetic screens to look at mutants and so on. And so how do you actually therefore get this perfect control, right? What is the genetic, but also now the mechanical interactions to give rise to this perfect control system? And so, of course, how do you understand control? Well, you try to break the control. Just like geneticists have done, you knock out the gene, you overexpress the gene. So we're now trying to overexpose the larvae to extra external mechanical forces to look at the consequence of that. And so now what we do to do that is, instead of having these straight channels where the larvae are quite happily crawling backwards and forwards, now we have these tight constrictions every few millimeters. And as you'll see here, this is basically two wing discs. This is the left and right. I actually don't know which one it is. But anyway, this one here, you'll see the larvae is trying to crawl through this very tight constriction. And as it does so, this wing disc is being stretched more than double the time, more than double the extent, right? Once it passes through, it goes back to normal again. So this kind of takes sometimes a few minutes to take place. And you can see the larvae really struggling to cross the channel. And as it's doing that, the wing discs are kind of being massaged. And eventually, boom, it kind of pumps through and goes through. And so what we've now done is to scale this up. So we've got literally hundreds of these PDMS printed channels. We allow these larvae to crawl backwards and forwards for about 4 hours. Pretty much after 4 hours, they stop. They get tired. We don't really know why. They get a bit lazy. And then we take them out. So what we do is we do automatic tracking. So every larva is an individual data point because they have different histories, and we track them. We record their history, their passage through these channels, and then we collect them, give each larva a unique ID. We then let them develop normally, and we let the wings hatch, or the adult hatch, and we look at phenotypes. So now we're just focusing on the wing phenotypes, but there are also phenotypes in different bits of the body. And so, example of some, I'm sure we're amongst lots of fly friends here. You probably can tell me better what phenotypes they may represent, at least classical genetic phenotypes they may represent. So for sure, a notch. Agree, Sarah? Maybe, I don't know. But we do also see leg disc or leg defects, in fact, and it's always the same leg. It's really interesting. It's always L3. That's the closest to the wing disc in the kind of anatomy of the larvae. So lots of cool phenotypes. But remember, none of these were genetic mutants. They were all supposedly wild type, but we mechanically challenged them. Yet, the phenotypes that are popping out are classical genetic mutants of classical pathways that we all know and love. And so also what's amazing is with these phenotypes, for example, if we drive RNAi just in the wing disc of a certain mechanical gene, for example, softening it by removing a little bit of myosin, then these phenotypes increase in propensity by a lot. And so these quantitative numbers we're still quantifying, but essentially what we can do is to use this system to make a genetic and a mechanic interaction modifier screen. So my PhD was pretty much a modifier screen. My supervisor's right there, Matthew. And I remember at the time I said, I'm never going to ever do a screen ever again. But here we are doing a genetic and mechanics modifying screen. And in fact, we're doing this in China, and I'm going on Friday. So we've designed a summer school in China. It's a joint summer school between Tsinghua and UCL. 3 weeks, the students will be passaging these larvae in these channels and collecting hundreds, thousands, hopefully, of data points for us to analyze together. And so I'm really excited by this large-scale batch of data because it's still fairly slow if we do it one by one and postdoc at a time in the lab. And so maybe in a few weeks we can tell you a bit more about that. So then finally, this is where I think that my analogy is, and again, my architect husband helped me with this, with these movies. So I guess in terms of let's say, intrinsic control, genetic control, we generally think of it as this, in the sense that, you know, the rods and the connections could represent genes or gene interactions, right? And obviously, if you break each of these, if you take apart each of these connections, your shape is going to collapse, your building's collapsing, right? But also, there's another way to collapse the building, which is to apply a external mechanical force. So things fall apart, but, you know, the individual, oops, sorry, I clearly didn't rehearse this enough. So genetic intrinsic mutations, right? You're breaking each of these rods, but then here you're slowly applying an external force. The connection is still maintained, but the structure still breaks or deforms. However, you can also have situations where you apply this external force, but it can also trigger some sticks to break first. Right, so I think that's what we're trying to understand here. I'm not trying to find the next mechanosensitive pathway, but I'm trying to find which ones or which genes or interactions are more mechanosensitive than others. And this, I think, is the really analogy we're trying to figure out with this kind of new screen that we're doing. Okay, and so this is where I would normally go to David and really, you know, try to write a grant and ask for his feedback. And this is 2016, and, you know, don't read all the details, it's really embarrassing, but he would always be, so I basically always asked David to read my grants and my papers, and he'd be always very critical, but also very supportive and very positive in the sense that, okay, it's cool, it's crazy, but you got to do this, or don't do this. And we heard from Caroline, you know, yeah, don't say interestingly or importantly, let them judge for themselves, let the reader judge. And also avoid polite English understatements, which yes, you know. So yeah, I mean, now, yes, I think David would have loved this project. Yeah, it's a shame I can't, you know, brainstorm with him, and yeah, like someone else, I don't know what to do next. So we'll see. I just have to come up with the next path myself, I guess. And yes, so that's all, really. Yeah, I miss David a lot.
Thank you very much, Yanlan. That was great. Maybe we have to have one burning question if there is one? Okay, so that's fantastic. Thank you. And then we'll move on to our last